speaker on the list is Leah Broekgaarden from the Dutch Neuromuscular Disease Organization. It's up to you.
Thank you. Well, good afternoon, everybody. First of all, I would like to thank Tiffany, Randall, and Marilyn for inviting me. It's the second time I'm in San Antonio, but I have the privilege to have been part of the IPA for several years. And the first moment I met Randall was in 1998, I guess, when we went out to have something to eat near the canal in the place where I live. It was snowing at that moment, freezing cold, but since then we've built up a friendship which is really very valuable for me. I'm very proud and it's a privilege for me to be part of the Pompe community, not only in the Netherlands but also internationally. Well, I may start with a kickoff of this newborn screening part of the program, so I thought it would be nice to start with a small YouTube film, and proactively I've asked Thomas Schaller to sit here and to help me. He's an IPA board member, so whenever it doesn't function, he'll help me. This is okay. Make it a little bit bigger. Take some time to upload it. So just to set your mind a little bit, newborns, it will start in a moment. Not in the meanwhile, I can go on talking. There are a lot of people here who are directly in contact with other Pompe patients around the world. We welcome people from Hong Kong, New Zealand. Well, I'm giving this presentation because Stephanie Wijnreich, who is doing this survey, couldn't come over here. She's working in my department at the VSN as well, working for Arnold Reuser. Newborn screening, a questionnaire study, that's what it's all about. Well, our health council of the Netherlands is talking about newborn screening when you talk about newborn screening shortly after birth. And it should have obvious benefits. A direct benefit for the child, if screening prevents major irreversible health damage, and indirectly if screening leads to improvement in the diagnostic of or the care. Our health council in the Netherlands is using three categories. Category 1 is yes, it's an uptake in our newborn screening program, like for PKU. And category 2, there is insufficient evidence, like still it's there for Pompe disease, so we have to lobby. And category 3, it's no, there's a no, like still there is a no for Duchenne muscular dystrophy in the Netherlands. Well, a lobby in the Netherlands, that's not that difficult. We are quite a small country, and I can tell you that we lobby a lot. Our Minister of Health is living in the same town I'm living in, so I meet her quite frequently at the butcher store. So it's not too difficult to discuss things with her. And also Rotterdam, because we are not going to lobby for newborn screening without our Rotterdam Erasmus Pompe team. But it's only 1 hour from the place I'm working and living. We looked at a review from the American College of Medical Geneticists, and that was really helpful, and that triggered us because they say that there are no data available regarding the acceptability to consumers of screening, and this is especially important because of the potential to diagnose late onset disease. And when you talk about late onset disease, we know there are several issues. Positive ones, because in unstolders and other stolders today, early treatment in time, it's enormous important. And also you don't want a diagnostic delay or a wrong diagnosis, as we now see quite frequently that people get a limb-girdle diagnosis in some countries while they have got Pompe disease. But we also know that there could be financial issues if people do get the late onset Pompe diagnosis. Maybe insurance companies will tell them, well, we are not going to insure you. You can't get money to buy a house or whatever. And the other aspect could be that people could feel burdened by the knowledge of the diagnosis. And there just was an article in Neurology about it. I'm fine, I'm just waiting for my disease. because we know that Pompe disease can be clinically silent for decades. There will be, in the upcoming period, a growing group of people who are patients in waiting, and something we should be aware of. We also know, and that's why I put the picture in it, that the future perspective will change if you know you've got late onset Pompe disease. On the other hand, we also know that there will be a lot of new therapies on the market in 10 or 20 years. So maybe then there will be a moment where no one will be confronted with the symptoms. That's what we all hope and pray for. Well, once again, we were triggered by this question, this remark. We need to know more about acceptability of consumers. So that's why, knowing the problem, we thought, well, we should start a project. And that's what we did. We started a project, within the project, Sustainable Orphan Drug Development through Registries and Monitoring. It's being worked out under the umbrella of Top Institute Pharma in Leiden, the Netherlands. And what's being studied is the acceptability of the screening to the public and the parents and the patients. There are also other items involved, but we are especially dedicated and putting our energy into the acceptability of screening to the public. And it's done in a questionnaire study. Key features: high school reading level, and there was an introduction on Pompe disease and newborn screening, written, visual and numerical. 3 scenarios were put forward to the people about positive test results concerning the classical infantile, false positive and later onset form. Method and questions, well, it was a survey, a simple survey for consumers and parents. Do they support the introduction of this newborn screening? And what is their moral reasoning about the dilemmas of the screening? And do they accept the expected rate of false positives? And how do they feel about detection at birth of disease, which may not manifest symptoms till much later in life? This is what we ask them. We ask them these questions, and I'm coming to the results later on, because I can't say too much about it yet. But I think it's also important to know a little bit more about neonatal screening, newborn screening, and the opinion in other countries. And like I told you before, there are people from Italy, UK, New Zealand, Germany, the Netherlands, United States, I forget, people from Hong Kong, all in this audience, and they all have an opinion, I think, and there are also timelines in their mind. I've put this clock, this picture of the clock over here because I hoped that the representatives from Russia would be present. It's a clock from Mr. Sokolov, made in St. Petersburg, and I thought it would be a nice bridge to introduce them, but they couldn't come, sadly enough. But once again, I hope that after this session there will be discussion, and that people from all of these countries will come forward with their ideas and their timelines. The results concerning this study are not published yet, and I promise you, when the publications are there, it will be put on the wonderful IPA website and on the wonderful AMDA website, so that it's available for all of you. And we are all in favor, I guess, but I don't know yet, about neonatal screening. But when we start neonatal screening, I think what we need is the best test available. You all are aware of Michelin stars. I think we need 5 pluses for a test. It should be the best one. But also when you start newborn screening in a country, the drug or the treatment should be available, as well as reimbursement. And you need real good expert centers and psychosocial guidance for all people, for the parents with newborn children, but also for the people who get their diagnosis and know that they are a late onset patient. And we need a toolkit. In this we can learn from cystic fibrosis, for example. They made a wonderful toolkit with information in it and guidelines in it. And I think the patient organizations in every country, and also the IPA, should try and put all their efforts and ideas in it to help to make it a really suitable toolkit, not only for the experts and the specialists in the field, but also for the patients who in the end are the ones where it's all about. This was my last slide. I only wanted to pinpoint the topic. There are a lot of contributors to this questionnaire study. Of course, the Erasmus University, but also 2 other universities in the Netherlands, one in Amsterdam and the other one in Maastricht. It has been done with the cooperation of the Dutch Association for Neuromuscular Diseases, where we have an AMDA POMPE group in it, with the Netherlands Institute for Health Service Research and lay readers. And it was funded through the Top Institute Pharma project. Thank you very much.
Professor Y.T. Chen from Duke University of North Carolina. They know you because you applaud beforehand. Very good.
No, because I'm old.
Wait a minute, we'll try that out. Who is the oldest? I don't know.
Well, anyway, I'm very happy to be here. And in the past several years, actually, I stationed in Taiwan most of the time and not able to attend many of these Pompey meetings. So really glad to have the opportunity to see many old friends and meet lots of new friends and patients and families. Arno and I and Paul, I think we are the 3 oldest people doing the Pompey research. But I'm very, very happy. This morning we have a lot of the new generation therapy, but also we have a lot of the new doctors, scientists, and healthcare taker to help our patient and family with Pompe disease. So I think the lab goes on when we are reaching retirement age, but we have a new generation coming. And actually it's a many exciting ongoing research to better take care of patients with Pompe disease. I will present to you the newborn screening program in Taiwan, but this work primarily was done by Paul Hu and Nancy Chen from National Taiwan University Hospital. I want to bring back to the 1999 when we started the first human clinical trial in US using the CHO-produce enzyme. And that time we only have 3 babies enrolled in this first clinical trial. And showing here is 2 patients that enrolled in this clinical trial. And you can see here one patient is doing much better than the others. If you look this is a motor development milestone, and you can see 2 patients, the first and the second patient actually did not doing really well, but the 3rd patient actually grow normally. A lot of difference at that time we can think of. The one thing is the antibodies and the cream status. But when we published at that time, nobody believed it. But we also thought it's maybe due to the age and onset of therapy and also severity of the disease, because this patient was treated earlier, but just a couple months earlier than the other patient. Then, when there are more and more patients receiving the myelin therapy around the world, then it becomes very clear. Some patients doing better than the others. So some patients actually look very normal, and some patients still require ventilatory support. And all these are the infantile patients. So even within the infantile patient, there are patients respond much better than others. So the variability in response to enzyme replacement therapy, well, we also know the heart respond better than skeletal muscle. Why do some patients respond better? And there are potential factors I list here. Age and onset of therapy, severity of the disease, and this has is very relevant to do when the disease are diagnosed. The dose, tissue delivery, muscle fiber type, and the cream status and antibody. The antibody Dr. Kishnani is going to address how we manage these rising antibodies tomorrow. And Dr. Raben is talking about the autophagic problem in Pompey disease. But I want to address these two questions, age at onset of therapy and severity of the disease. And we know this really depends on when the diagnosis is made. So we want to push the early diagnosis as possible. So the earliest you can do the diagnosis, of course, I mean, you can do the prenatal, but you need to have a family history. So we decided in Taiwan to push the early diagnosis at newborn period. So the pilot program started October 2005. Started with single hospital, then graduated in 2008. We reached to many hospitals. So during this period, from about 4 years period, the total newborn screen is more than 300,000, 344,000. And this is from the heel stick from the baby before they go home. So the first screen that the 0.6% was considered was abnormal. And within this almost 2,000 patients, there are 17 is highly abnormal. And these are the patients we record immediately, and we call the patient back, get the blood and fibroblast assay and activities. And within this 17 patients, actually 6 is diagnosed as infantile onset Pompey disease by the chest X-ray, the EKG, and so on. And 11 without the cardiomegaly and was considered as suspicious later onset Pompey disease, the NBSL here. And for this patients are abnormal, but not as very highly abnormal, that when we repeat the blood, eventually it's still abnormal. There's 185 patients. And these 185 patients was recorded and draw the blood and skin biopsy and get inactivity. And within this 185, actually 2 with inactivity less than 5% normal mean turn out to be the suspicious later onset Pompey disease. And over here, this actually now many of these patients we already have mutational analysis, and they turn out to be either carrier or patient with we call pseudo deficiency. But their inactivity actually is greater than 5%. So we still able to tell by inactivity level, at least in Chinese population, that if less than 5 is either infantile onset Pompey or suspicious later onset Pompey disease. So I want to tell you, so these 6 newborn baby diagnosed with infantile onset Pompe disease that was all received the enzyme replacement therapy within the first month of life. Before that. So the overall incidence of Pompe disease in Taiwan is approximately 1 in 18,000, and for infantile onset is 1 in 57,000, and for later onset is 1 in 26,000. Okay, so those 6 newborn identified with infantile onset Pompe disease was put on the treatment within the first month of life. And here we compare two. The red color is untreated patient, and the blue color is patient diagnosed, the infantile Pompe patient diagnosed clinically. And the green color is a newborn that diagnosed with newborn screening and was put on early treatment. So you can see here that in the clinical diagnosed patient, although they may be 3 or 4 months apart from the newborn screening, but there's a decrease of the survival. And half of them, even with enzyme replacement therapy, still required ventilator support. In contrast, those 6 babies that identified through newborn screening and put on the treatment are doing very well and continue to do well. Look for the chest X-ray. This is a 3 newborn diagnosed through newborn screening with a very big heart and with treatment, and you can see the decrease of the heart size. And clinically, you can also measure the left ventricular max index, and you can see all this patient with decreased size with treatment. For this motor development, that all these 6 patients actually has near normal motor outcome and continue to improve. Well, this is another way to look for the motor development is to see the time they walk independently. And again, this is none of the untreated patient able to walk, and only about 40% of the treated patient but diagnosed clinically, not by through newborn screening, that only 40% able to reach walk independently. And all of the 6 newborn infantile Pompe patients able to achieve the walk independently. This is one of the patients showing that even at the newborn period already has very significant the damage to the muscle. And with treatment, it's a complete reverse. Okay, so I just give you briefly summary for this infantile onset Pompe disease. If you diagnose it early and treat it within the first of months, actually the outcome is really, really good. The question is really this later onset Pompe disease patient. There's 2 here and 11 here. So these 13 patients. And this 13 patients, actually we all have mutation analysis, and indeed they carry this, the Pompe mutation. But before we talk about how we are going to treat these 13 patients, decision to treat or not is very, very difficult. But I want to show you in Chinese later onset Pompe disease patient, actually a little bit different from Caucasian patient, from a patient in US and in Europe. The disease, the symptom of onset is the median age is 9, range of 1 to 16. But usually the diagnosis was not made until much later. It's about 21. But you can see there's a very rapid downhill, the downhill course of the disease, and the median age for ventilator is 22. And this onset age, I know in the German publication and also the Dutch publication, usually is about 20 years when they have a symptom. So in Chinese population, it's actually very, very, it's actually earlier onset. And the diagnosis is not so easy, but usually at the time of diagnosis, they already showing the decline, and lots of patients actually on ventilator. So the decision whether to treat those 13 patients we identified through newborn screening actually is very, very difficult. We know the Chinese patient actually has a rapid progressively course of the disease, but we still don't know when we should to start the treatment. The mutation identified is actually one of the mutation is D645E and with pseudo deficiency. Another mutation is another point mutation shown here. But I want to point out that this is also very different from Caucasian population, because Caucasian population for the late onset Pompey disease is one of the splicing mutation in the intron. And in Chinese patient, we don't have this mutation. So the question is, now we can early detect those later onset. Shall we start the treatment? And as I mentioned, it's a difficult clinical decision. Some of the newborn with later onset Pompey disease, usually this is within 3 months of age. Actually, some already, this is later onset, already has high CK. And some with consistent high CK with follow-up. Okay? So these are the patients we think is probably worthwhile for the treatment. But also we consider some newborn with later onset Pompey disease have motor problem. Primarily is the gross motors, not so much about this fine motor. So some of the patients with gross motor delayed, then we consider for treatment. But again, this is not absolutely. So currently, up to last month, a total of 10 newborn with infantile onset Pompey disease, and these 10 all treated immediately. And Dr. Wu started with 20 milligram per kilo every other week. And another thing I didn't mention is all the Chinese patients actually is cream positive. And very, very few I know of have antibody problem. So this is more clean cut. All those 10 patients diagnosed as a newborn was treated. 16 patients suspect to have a later onset Pompey disease. And all these 16 patients confirmed by fibroblast inactivity and also by mutation analysis. And so far, Dr. Hu treated 6 of these 16 patients. And the criteria for the decision for treat or not is actually is not very stringent, and more of it depend on the persistent high CK or the motor developmental delay or some muscle weakness. So conclusion, I think for the newborn screening, for the infantile onset Pompey disease, certainly early diagnosis and early ERT have improved the outcomes of the infantile onset Pompey disease. And for the later onset, of course, early diagnosis allowed the early treatment. But when to treat, we don't know yet. But the early treatment is probably important given Chinese patients have a rapid progressive course of the disease. But we still need a lot of long-term follow-up data. I think I stop here. Oh, I just want to show you, this is the Pompey patient family reunion. All this here, these are actually all this screened from newborn. From newborn screening is about five years old. And this is clinical diagnosed patient started at 4 months of age and did not respond that well. I want to show you the last slide. This was a couple of weeks ago. We had a Southeast Asian Pompey family gathering. They are family from Hong Kong, from Singapore, and from Malaysia. And one of them, and Mr. Lai, Terry Lai, actually is here, representative from Hong Kong. Many of them, they're not able to come here, but they want me to show you this slide, and they are with you. Thank you. Thank you very much for your attention.
The title about Pompey disease and the different ways of diagnosing the disease. All of you have been diagnosed with the disease, and you probably kind of know how the procedures went. Maybe you were diagnosed on blood, white blood cells maybe, or directly on DNA, or maybe a blood spot, or maybe skin fibroblasts. Different laboratories use different technologies, and most laboratories know what they are doing. So at the end, they are supposed to come with the right diagnosis, but sometimes it can be more difficult than you think it should be. This is the way. Oh, I don't. Does it work with my finger? Yeah, it does. Okay. So we are set up. Of course, first of all, you have the clinical aspects. All right. So where we are, we have the clinical aspects, and in our center, of course, that's Ans van der Ploeg. For the children, and Peter van Doorn, he's a neurologist for the adults. And then in the Department of Clinical Genetics, we have a kind of split up in different activities. We have the DNA analysis with a supervisor, Holly, and we have the prenatal enzymology, which is Verheyen, and then you have the metabolites. Think about CLUG4 marker in the urine. And then we have research development in a separate setting, because in the diagnostic setting it's all protocolized, and we do not exchange any material, no names, no nothing, so it makes it very complicated, but it secures privacy. I go now page up, page down.
Next time, page down.
Thanks. So we had this very, very happy event last year. A couple with a newborn and their first child, and the lady has Pompey's disease, and when she got the first child she was 42. She was at enzyme replacement therapy for more than a year, and during the pregnancy the therapy was continued, and of course a lot of care was taken for the child, etc., but it was a happy ending. And this paper is now in press, or maybe already published very recently. So it is the first experience with enzyme replacement therapy during pregnancy and lactation in Pompe disease. And why lactation? Because it's not only that you give the enzyme to the mother while she's pregnant, so the enzyme may reach the fetus, but also then we thought, okay, if the mother is feeding the baby, is there any enzyme in the milk? And to our big surprise, indeed, if you give an infusion intravenously, some of the enzyme comes out in the breast milk. All right, page down. Then since I'm a biochemist, you know, I'm interested in things. So this man, the father, I've taken the father, maybe his weight is 80 kilograms. So that means he consists of 100 million times a million cells. And a cells is the unit of a tissue, the unit of an organ. You have cells that make the brain, you have cells that make the muscle, cells that make the teeth or the eyes. So all different types of cells, and that makes the body. They go all different directions. 100 million times a million. The child, what's the weight of a child? 4 kilograms. So 5% of that number of cells. And then you've already seen this one. It's beautiful. Life is so beautiful. Because it starts with this single, and now it's no longer single. I want to have it back single. This is a fertilized oocyte. Damn it. You know, these new computers, they are so fast. Okay, a fertilized oocyte. How do you know it's fertilized? It's just coming from the ovaria. You can see that because all the other little things around it also are cells, and these are the supporting cells. So during ovulation, the oocyte comes out, boom. But around it is this whole, what you call, it's a corona of cells, and they support the egg, they feed the egg, and then when the egg is fertilized by the sperm, you have these two little things sitting here. And it's called a polar body because it's sitting on the pole, like a globe. It's sitting on the pole. And what is it actually? Females, they have 2 X chromosomes, XX, and males have XY. In the germ cells, you need to combine. So you have 1 X from the female and 1 X from the male, makes it XX, the female. If you have an X from the female and a Y, makes a male. But the female has 2 X's. So one and only one has to be active. You can't have a female with 2 X's. So the one goes out. So when the sperm comes in, one of the X's goes out. And this is one of the X's that comes out. Okay, now I have the next one. It's the movie. You have to watch very carefully because you can see the X come out. Okay, watch it. Now page down. Up, down. Okay, this is just the beginning of the movie. All right, now watch here. This is the oocyte. This is one, there it is. So the X came out. So that means the sperm went in. And then immediately the cell starts to divide. So you have 1, you have 2, you have 4, bloop, bloop, bloop. You make 8. Yeah, that's how it started with all of us. 8, bloop, bloop, more bloop, bloops. And then you have 16, 31. And then they're all the same still. If you would split it, you would have 2 embryos. But now it starts to differentiate, because at the one side you get this empty space. This part is becoming the placenta, and this part is becoming the embryo. Okay. Now, why all this? Why all this? Here you see the same. You have 1, you have the 2 polar bodies, you get 2, you get 4. Inside every cell, you have the nucleus. Okay, and the nucleus contains the DNA. And the DNA is the genetic information. And you need this information to make the cell and to make all the proteins inside the cells. To make the nucleus, to make all the other organelles, including the lysosomes. And also you need the genetic information to make acid alpha-glucosidase, GAA. So the information is in the nucleus. And it's there in the form, as we all know, it's DNA. And what is DNA? DNA is a chemical structure that is so clever that it can contain information in a genetic code. And the genetic code only consists of four letters. So you have an A, wait a minute, you have an A or a T, a G or a C, so only four. But you can make endless combinations, because it can be ATGC, it can be AGCT, and this goes on and on and on and on and on and on. No more than that, it's simply the information. So you have another big figure, 3,000 million of these letters. And altogether it's about 25,000 genes that can make 25,000 products. So this is the information now, this is the DNA. And if you want to use the information, you have to open up the DNA. DNA is a double strand, so it opens up. And of course, if you want to make sure that you don't get any mistake in this DNA, you're not going to use it to make proteins. No, first you copy it. You make copies. And the original goes back into the drawer, you don't touch it, and then you start to work with the copies. And the copies are the RNA. They too consist of four letters, the same, and they have all the genetic information, and based on the order of the letters, you get a protein. And the protein is a completely different chemical structure. Has nothing to do with DNA, nothing to do. Has completely different function. And how do you make a protein? A protein consists of amino acids. So you have this one that looks like a jelly bean, and this one has a different shape. You know, this mouse, okay, has a different shape. So now you line up all these amino acids and you do it in a certain order. And that is how you can make zillions of different proteins. Each time you take another amino acid in another order. And this is the way you can build acid alpha-glucosidase. If you have 952 amino acids in the right order, you have functional alpha-glucosidase. If one of them is missing or in the wrong order, you have a problem. Okay, and this very long chain is then being folded. So this is the folded structure of acid alpha-glucosidase. And this morning you heard about the anchors at the sites to grab the mannose-6-phosphate receptor. They're sitting here, one there, the N, one here, one there, 7 in total. So the anchors, indeed, they sit on the outside. And the glycogen that needs to be degraded goes in here, in this pocket. So the protein binds to the glycogen and then takes off a glucose, another one, another one, another one, another one, and that's the way you degrade your glycogen. Then this is the total picture. You have a cell, so one of these so many million. Every cell, of course, needs to have the same genetic information. That's the DNA. Based on this genetic information, you can produce acid alpha-glucosidase, GAA. Acid alpha-glucosidase needs to go to the right compartment, and we all know the right compartment is the lysosome. And inside the lysosome, it needs to degrade glycogen, this is this structure, into glucose. And the glycogen comes from here, and it enters by autophagy. Much more about autophagy tomorrow. So if this protein, if this enzyme is not working, you accumulate glycogen. All right, then how can you diagnose the disease? You diagnose at different levels. You can look at the order of the DNA, the sequence, and if any letter is missing or wrong, replaced or whatever, you have diagnosed the disease. But of course you can also look whether glycogen accumulates, that's another method, or you can measure the activity of acid alpha-glucosidase. And you can measure the activity of acid alpha-glucosidase in white blood cells, or in a blood spot, or in cultured fibroblasts. So there are different ways and different methodologies. All right, now I'm going to skip all this maybe I should show this, because we keep a mutation database at the internet, and we try to keep it updated in the sense that we try to list all the different abnormalities in the DNA of patients with Pompe disease. And patients are following our activities, and once in a while I get an email message from a patient who tells me, Dr. Reuser, you've missed a mutation in your database. Fantastic. And then we correct, and we try to be updated. So if anyone finds mistakes, please, yes, send an email, and we try to correct. I'm not going to do all this, and I'm not going to do this either. The only thing I want to show is we have also tested whether newborn screening would be technically feasible in blood spots. And I think we can confirm the message we just received from Dr. YT Chen that yes, it can be done. And indeed, it's easier in the Caucasian population than it is in the Asian population. But technically, there's no problem. It can be done. And the question raised by Dr. Chen, should we do it? They already decided, yes, it should be done. But in other countries, there may be another way of decision making. Going to skip this. I was invited 3 years ago now to a meeting of Shire LSD Symposium in Lisbon. And this was a proposition. It was not my idea. They made the proposition. This house believes newborn screening for LSD should be obligatory. Obligatory, yeah. And that's a very strong statement. It just means you have to come in with your child because it's obligatory. Obligatory is the word, isn't it? Obligatory. I knew there was something wrong. It didn't sound quite right. Obligatory. And I had to defend this proposition. So before I said, I can't defend, this is impossible because you cannot, never, ever. But said, yeah, but of course no one else wants to defend, so do you want to defend? So I did my thing. And I had some arguments, and these are the arguments, and I use them now for the discussion that we can have in 2 more slides. Of course, why would you do screening? We have learned that symptoms are preceded by tissue and organ damage. You don't see so much, but if you make an X-ray, you do see the cardiomegaly. The diagnosis is often delayed. Most of you will know that. So if you diagnose immediately after birth, you're gaining years and years and years. Patients benefit from early diagnosis through optimal care, yes, and therapy, yes, if available. And for the time being, that is enzyme replacement therapy, maybe in combination with chaperone, as we have heard this morning, and maybe in future, the gene therapy. Screening offers benefits to family members, and that is also an important issue, because sometimes patients are diagnosed late, and you have more affected siblings in the same family. And maybe parents would have decided not to have any more children if they had known. So this is, for the counseling, an important aspect. Newborn screening, as I said, provides the key to genetic counseling. You can warn the parents. Couples appreciate to know their genetic risks, as always. They said, if I had known, I could have taken my own decision. Newborn screening provides the key to building natural history profiles, because you know very rapidly if a child is going to be affected and when to start treatment. We have just heard the example, the later onset Pompe disease in Taiwan. If you don't know, you don't do anything. If you know, you can take a decision. This patient, yes, this patient, no. And of course, by doing all this, we will learn a lot about the genotype-phenotype correlation. And if we have some more knowledge about genotype-phenotype correlations, it's easier to have a prognostic advice to the parents based on the genotype. This I took from a newspaper. It is about Sergey Brin. They screened his whole genome, because that can now be done. Historically, it took us 2 years to diagnose the first Pompe disease patients on the level of DNA. 2 years for a single patient. You can now do the whole genome in 1 week for a fraction of the costs. So time is changing. And then, of course, if you screen the whole genome, this Google founder, Sergey Brin, he is genetically predisposed to getting Parkinson's disease because you can read it from his DNA. And then what would you do? You like to know or you don't like to know? And this is the opinion of Sergey Brin. He says, I know early in my life something I am substantially predisposed to. I have the opportunity to adjust my life. I also have the opportunity to support research into this disease long before it affects me. And as I said, the DNA screening is going to become very cheap. Okay, this house believes that was the proposition. But first of all, I think that the newborn screening should be to the benefit of the patient. And to the benefit of the patient, again, in my collection, I found this slide. It was from the very first brochure of the AMDA. And on the back flap, it was asking for a donation because of this baby who had the very severe form of infantile Pompe disease long before the treatment was started. And to end, I have this movie. I got it from Ans van der Pleuk about a patient, infantile onset Pompe disease, who was treated, under treatment, and she's doing reasonably well. To this. Yeah, all right. So I leave this on for the discussion. So challenges now for newborn screening, how to provide an even better treatment. Should that be through newborn screening or just through developing new technologies? And if you use the newborn screening, how to predict the clinical outcome and when to start treatment. So with this.
But I remember as far back as 3rd grade, I wasn't able to do a sit-up, and we had an event at school. It was called Field Day, and I remember telling my mother that I couldn't go to school that day because I had a headache. I lied basically, but I mean I was self-conscious about it even in 3rd grade, so I knew there was something wrong, and I always suspected that these markers like CK, which is very sensitive. I always suspected that was elevated. That was how it was identified, from elevated CK and liver enzymes. But I always suspected that they were elevated way before it was first discovered. And I was just curious of the 6 patients that were treated, the late onset patients. So were the patients that had the elevated CK were the same patients that had the developmental delays, or were these different?
Yeah, I think your experience, retrospect, you knew something was wrong where before you diagnosed. I think that's very, very typical for late onset patient. For those 6 patients we identified through the newborn screening and put on treatment, actually only 3 has persistent high CK, and the other 3, the CK was normal, but the doctor decided to treat primarily because of the motor delay. Yeah. So it's not all the patient has high CK. And we also know CK actually is highly variable in terms of your daily activities. So the answer to your question is those 6 patients, only half actually has high CK.
So why did you decide to, so 6 of them were treated?
But all of them, before the treatment, have a muscle biopsy, and all are normal.
Abnormal.
Yeah. So this is, Dr. also mentioned, the symptom precede the organ tissue damage. Although you don't have symptoms, but if you do the biopsy, yeah, most of the little ones already have pathology.
Yeah, it'll be interesting to see those patients 20 years from now to see how they.
Yes, exactly.
Yeah, thank you so much. Those are just fascinating presentations. Just to follow on to that question, Professor Chen, the CK, I'm just curious to know what cutoffs you have for CK before you begin to treat. Do you have like a cutoff, or is it in physician's decision as to what to treat? And for how long does it have to be elevated? Because I think, like you're saying, it changes from day to day and week to week. So what level do you use? Are you using the same cutoffs or not? And as a follow-on to that, I'm wondering whether urinary X4 features as well in your decision to begin to treat. And then just so that I don't miss this opportunity, Professor Rajat, the next question really I find so fascinating. Yes, sir. I'm asking 2 questions and I apologize. The next question I find so fascinating, the patient who had a baby on ERT and he said it crosses into milk. I imagine it crosses the placenta as well. And I don't know whether this is for you or whether maybe your Jensen colleagues could help us with this. If it's able to cross in utero, would that be a way of tricking the immune system into not producing antibodies against this later on? I don't know whether we have that information.
I'll try to give an answer, but maybe my T, if you think it's different, then maybe assist in my answer. First of all, I'm not so sure that it crosses the placenta. Because there was long time ago a story about a twin calf in Australia, and the one embryo was affected and the other one wasn't, and there was no cross correction. So from that we kind of decided that it would probably not cross the placental barrier. There was another incident in the laboratory where we were trying out the effects of enzyme replacement therapy, and one of the female mice was pregnant, and we didn't know. So when we noticed afterwards, we checked out the children, and since this was a knockout couple, so father and mother both had Pompey disease, we were checking out whether these newborn babies would now have enzyme activity, but they didn't. So also from that experiment we had no clue about exchange across the placental barrier. And how it precisely enters into the milk, I don't know. The mammary gland is a complicated gland. Of course there's a lot of blood. But the enzyme, of course, has to first leave the bloodstream across the capillary walls, and then goes through the tissue, and then creep out in between the cells of the skin, actually, because the glands are compatible with skin cells, so it kind of slips through and then enters into the milk. Immunoglobulins do so too. So maybe what happens in the mammary gland is not compatible with what's happened in the placenta. But I have a little evidence.
I can add a little to that, Alison. Yeah.
Where are you? Oh, Alison, hi. Yeah, please.
We've never looked to see if it was present in the milk, but when we did all the reproductive toxicology studies for myozyme, we did look in animals to see if it crossed the placenta, and it did not cross the placenta in either mice or in rabbits. And the way that we did this is we took a look at, because obviously the baby mice are so small, it was very difficult to sample them. But what we did is we looked in the livers of the pups, because we thought if myozyme was there, it would go to the liver, because we know so much of it goes to the liver. And we saw a really nice dose response in the mom's livers, and we also saw a nice dose response in the placenta, but we could see nothing at all in the livers of the pups. So from that we concluded, and we did that both in mice and rabbits, that it did not cross the placenta.
And then the other part of the question, I was thinking about what type of situation actually you depict, because then the mother was being treated. That was your situation. The mother was being treated, and then whether the child would be tolerant to.
The earlier you introduce it in the child, in the infant or in the fetus, the more likely you are to trick the immune system into not considering this as non-self. So I was wondering if now that it doesn't cross the placenta, I guess I have my answer, but I was wondering if it did, then would that be a way of just tricking the immune system into not producing antibodies later on when we begin to treat
the immune system of the newborn?
Yeah.
Yeah, but that means that then the proposition is that the newborn would be affected.
Yeah, because
that won't happen, that situation. I hope it will not happen, that the mother is a patient, and that even in that situation that the mother has a pregnancy with an affected child. You know, you really try to prevent that. But okay, it's a theoretical thing.
I just want to add that we do have a sub-registry for both pregnancy and lactation in women with Pompe disease. So if anyone's interested, they can contact anyone with Genzyme and can get them to the right person. So, and we'll also have some external investigators that are going to be collecting data around all pregnancies in women, whether they were or currently are on enzyme replacement therapy, just to try and gather data around that.
Anything known? How many situations at present in the registry?
An exceptionally small number. We just don't have the data in there yet.
Okay.
We're hoping that this new effort that we have some external folks working on is going to help us get some data, but we do have a small amount of data, and we'll publish what we have as soon as we have one more.
Thank you. More questions?
I just had one. Brian's next, actually. I just had a quick observation that here in the United States we have the HHS Advisory Committee that sort of makes the decisions nationally for the different conditions that get tested for newborn screening. And Pompey was up for nomination, I guess it was 3 years ago, and it was declined because of the high false positives, I guess, and then the late onset issue. But it seems that Rodney Howe, who's the chairman of that committee, recently said that Pompey should be renominated, and that he would, I don't know if he said he would support it, but he was the one that said it should be renominated. So I think it was pretty nice to have the chairman of that committee on our side.
Thanks. Yeah, question?
Well, I guess mine's more of a comment, and I don't want to be a downer, but I think it is a slippery slope to the late onset. I know Jared's situation was one. My symptoms didn't come on until I was about 36, 37, and just looking back, I'm not sure I really would have wanted to know. Had that hang kind of over my life for those first 36 years. I mean, I guess theoretically it would have helped with the diagnosis, and I could have started treatment a little sooner, but it's a tricky issue.
Dr. Kishnani, I think you still had a comment on the former question.
So this is George in response to, I was the one who actually submitted it for nomination for newborn screening for the US. And I think in retrospect, as I look at it, we were the first, so I think we were almost like the pilot scapegoat group. And at the time, there was a new set of rules that were being instituted by HRSA, and we were told that it required a US pilot before it could be put on the screening panel nationally. I think also that the data from Taiwan, and Dr. Chen can attest to that, it wasn't a higher false positive rate, it was rather a higher cutoff, which was deliberate, so that they wouldn't miss any cases. When Paul Hu and the group actually brought the cutoff down, there was no difference in the false positive rate as compared to others. So I think, yes, it does need to be revisited, and this is something that I am currently working with the college and with HRSA. And Rodney actually has stepped down now as the chair, Dr. Howell. But there are many on that committee who do believe that this is prime time for Pompey. The question is what technology, and just to put it in perspective, there are some states where newborn screening has started in the US for Pompey. So I'm hoping that with some of that data we can go back.
May I ask a question for Dr. Chen?
Sure.
Do you have any thoughts about the economics of the newborn screening? Or to me, it seems like it would. I mean, I don't care about the money really, but I know how the world works. It seems like it would be a possible cost savings measure in the long run. I don't know. Do you have any thoughts on the economics?
Well,
This is a very tough question. I mean, for the screening for Pompey, I know it's less than $10 US dollar. So that is not a problem. Really the problem is when you start the ERP, that is expensive. And but in Taiwan is national health insurance coverage. So basically it's a government pay for it. Yeah, but we also know we cannot put everybody on treatment because it's going to bankrupt on the government insurance. Yeah. So I think it's to me, if it is the therapy is cheap enough, I would certainly like to put everybody on it. Yeah, but right now it's not.
Dr. Chen, this is Marcus, just to make sure that you get an opportunity to answer. I asked a question about CK earlier on. Thank you.
I'm sorry. Yeah, the 6 late onset patient on treatment, 3 has CK. But CK is not the only criteria that the doctor decided to put on therapy. Because this patient also has hypotonia, muscle weakness, motor developmental delay, plus half of them has high CK. So CK is not the only criteria they will put on therapy. And the hex score, I think it's Dr. Kishnani, you have those data, the hex score?
Yes.
You want to comment?
Yes. Do I need a mic or?
yeah, sure. Mike's coming.
So I think.
Push it.
This is.
So we do have data, and this is in collaboration with the group in Taiwan, on the results of HEX4 both in infantile and in later onset. I think we've looked at it for infantile, and it appears that some of the infants did have HEX4 in the normal range, but some of them already have elevations of HEX4 in what would be considered otherwise clinically pre-symptomatic if a chest X-ray was not done. So I think HEX4 is going to be very useful. I think the utility of HEX4 in later onset is a question that you have. I think for one thing is that at Duke, this work was done by Sarah Young and by David Middington. I think we don't have appropriate cutoffs for HEX4, and now with the new cutoffs, what is the upper limits of normal? We are finding elevations in HEX4, but I think the issue that we have at hand is why do some late onset patients not have an increase in HEX4? I think it's probably related to the pathology, that it's not only the glycogen, there's also autophagy, but also are those the patients who are already too involved, where there's not enough glycogen to cause release of HEX4 in circulation. So my guess is that the later onset picked up early via newborn screening would actually have higher HEX4 than in the adults with later onset that are picked up. Let's see if this hypothesis comes true.
We have another question here.
Actually,
Sorry I let you wait so long.
Actually, it's not a question, it's a comment. The gentleman talked about would he have liked to known earlier about having the disease if he had an infantile study done or whatever, gene study. What do you want to know? I have a different perspective on that, in that I had a brother that had Pompey. It was 1972. He was quite a bit older than me. At the time, they thought it would show, the disease would show up at puberty. I was 9. I was the only one left in the family that hadn't reached puberty. They did the test, and I indeed had Pompey. My parents told me when I was 26. So at that point, you stand and you turn around and look at your life, and you go, had I known that, and the information was there, what would I have done different? What did I do in my life that hurt me? That that I could have avoided had my parents told me or looked out more for me. The damn presidential physical fitness test in middle school, 3 years running, the last one coming in on everything. They have a lot of psychological problems. And then when I got to college, I did the standard drinking thing. As every college student. Obviously, retrospect, a big no-no. So, there's more to it than just would I not want to know. There's a lot more involved in that. And for parents even to think about that my parents wanted me to grow up normal. That was their idea. Don't use it as an excuse. But I wasn't normal. They didn't do anything to protect me, if you will. I know that some of the teachers, I know for a fact that some of the teachers said I was lazy in gym class. No, I wasn't. I just couldn't do it. So, like I said, it has a lot of, it's not as simple as saying, would I know, would I do it, would I not do it? And then it gets later on, did I want to have kids? And all of that. And my wife, I had been engaged. She had a right to know what was coming. So it's not as simple as just, turning around and saying, I don't know if I want to know. It really, if you had known, there might have been a lot of things in your life you wouldn't have done, and it might not have kicked in as early, or you could have done some things to prevent it from, just a lot of different issues. It's just not that simple.
Thank you. Yes. Thank you very much for saying things. Because I think in the end these are the how shall I say this? These are the comments we need and the tips we need and the personal stories we need if we fill our toolkit with information, and if we decide to go and start with newborn screening, we have to get tips and experiences and put guidance in place for parents, people, patients, everyone. And then we need stories and expertise like yours. Thank you very much.
Okay, we're getting to that. There's one more.
We have time for about one more question. Yes.
Last question.
Yes.
And you have a comment to make then after?
Yes.
Yeah, okay. Yes, please. Oh, there you are.
I just have a comment. I know it's different for the late onset, but as the mother of a child with infant onset, to me, the main, if you have 5 infants, 50, 100, 200, when you look at the 100% of the 6 infants that are walking at this point, and we know without treatment or delayed treatment, you've got some that pass away before they can get enough treatment or not walk. That right there is the whole, that is the percentage you need right there to do newborn screening. There should be no other question whatsoever. Late onset, yes, I know it's some different, but even for late onset, I know there's a lot of things to look at, but by the time you're showing symptoms, you have damage. So, I know it's a money issue, starting treatment when you have that positive, but you're talking irreversible damage. So, as the mother of a child, you want to start within a few weeks if possible and have a better life for your child. A life, really, the only life.
Thank you for sharing this experience. This is bringing this afternoon to an end. Thanks again, all speakers, all day, all the people that were here. I think it was very successful. There's one other comment to be made by Andrea.
We are going to go ahead and wrap it up.
I want to thank you all for coming again, and I know I said this yesterday, but I'm really going to reiterate again how much I appreciate the fact that everybody is here, that we have so many patients, so many of the really experts in the field, and our industry partners all coming together. I think, we talked yesterday a few times about the Pompey model, and I think we should all take a moment to really reflect on how unique this is. This ability to come together, all three, really key partners in the treatment of Pompe disease, coming together in the same room and really having open and honest discussions. That doesn't happen for other diseases, and I think we're extremely lucky that we've been able to make that happen for Pompe. And I want to give a special thanks to all of our speakers who've come so very far and who are so extremely busy and who really go out of their way to be available to patients when they have questions. So can we give them all a round of applause, please? And with that, I want to go ahead and kick off the morning session. And in case anybody hasn't seen it, there is an AMDA table with items for sale. And I want to give a special shout out to Sharon Kruger, who wasn't able to be here, but she put together a high-protein, low-carb cookbook of her own recipes for patients, and we have that for sale if you're interested. So with that, let's get started.
All right. We have 3 sessions today. 2 more the scientific side, but still I think also for the patients of very great interest, because it's going to tell you a little bit where science is going and what to expect for the future. And thereafter we have a round table, and the round table will be led by Marise Schoneveld. The first session is called Future Research, and the first speaker is Dr. Nina Raben from NIH, and it is about autophagy. But yesterday we had dysphagia, difficulty swallowing, and this is autophagy, something with swallowing. She will explain.
Okay. I'd like to thank Tiffany for inviting me for this meeting. And I would also like to thank Priya Kishnani, YT Chen, and 2 doctors in Taiwan, Dr. Paul Hu and Nancy Xian, for providing muscle biopsies, which I will be showing today. And I have to say that the number of those biopsies which became available to me are not enough to come to a definite conclusion, so I will share some of the observations which I made. What? Okay. I will start by showing you several muscle biopsies from Pompe patients who have been on therapy for months and in some cases for several years. In all those cases, skeletal muscle in a biopsy is completely destroyed. Here is a case of a 7-year-old patient with infantile form of the disease on ERT since 5 months of age. And those empty spaces point to complete muscle destruction in this biopsy. Here is another way of analyzing muscle biopsy by looking at individual muscle fibers. So this image looks very colorful, but in fact, all those muscle fibers are completely destroyed because you do not see a striation pattern, which is so typical for normal muscle fibers. The same degree of muscle destruction you can see in these two biopsies from two cream-negative patients on therapy, one since 6 months of age and the second since 5 months of age. In this particular case, some of the muscle fibers are still partially preserved because you can see this typical striation pattern in some of them. And here is yet another way of looking at muscle biopsy by imaging the whole muscle biopsy. And this is a biopsy of an almost 5-year-old patient with infantile form of the disease on therapy since 7 months of age. You can see a single severely damaged fiber, which is still somewhat preserved, again, because you see this striation pattern. But what's striking about the whole biopsy that, again, all muscle fibers are completely destroyed. And the question is, how did it come to this? Despite the therapy. In other words, what is the chain of events which leads to this profound skeletal muscle destruction? And the traditional view of the pathogenesis of the disease, and the pathogenesis is exactly the chain of events which leads to organ dysfunction and the disease. And this traditional view was also shown yesterday, but in a different version of the cartoon. So the traditional view of the pathogenesis is that in Pompey disease, like in many other lysosomal storage diseases, the accumulation of undegraded substrate results in enlargement of the lysosomes, as shown here. In normal muscle, they look like little dots. So this by itself results in organ malfunction and the disease. The additional mechanism which is at play in Pompey skeletal muscle is that these lysosomes in muscle have very limited space where to expand. This leads to mechanical pressure, rupture of the lysosomes as shown here, and release of the accumulated substrate and also potentially toxic material into the cell, which leads to profound skeletal muscle destruction and end stage as shown here. Animal model is a very useful tool to follow the disease progression because you can look at how the disease develops at the level of individual cells. And what we found in our mouse model, that there was no stages described in a previous slide. What we see in our mouse model is indeed expanded lysosomes, as is typical for Pompey disease, shown here with red asterisks. But those lysosomes are quite well preserved. They are certainly not ruptured. And what we also see is a huge accumulation of undigested material, of undigested cellular debris, what is typically called autophagic accumulation. And this is shown by electron microscopy. What we also found was that this autophagic accumulation, or as we call it sometimes autophagic buildup, is found in virtually every muscle cell in a mouse model, and it spreads along the whole length of the fiber, as shown here by little bubbles. And it goes through the whole length of the fiber, and it often localized in the core of muscle fiber. So what is autophagy? Autophagy is one of the two major intracellular lysosome-dependent degradative pathway. It starts with the formation of double membrane, which engulfs part of the cell. The membrane closes, resulting in the formation of vesicles within the cell called autophagosome. Autophagosome fuse with lysosomes where the content of autophagosome is degraded and recycled. Fundamentally, this is a survival mechanism during starvation. So when the food is not available, the cell has to use its own resources to stay alive until the situation improves. However, it has been shown that even when the food surrounding the cell is available, the system still operates. It operates at relatively low level. And in this case, the function of the system is housekeeping function. The system allows the cells to rid itself of unwanted protein, protein aggregates, and damaged organelles, like, for example, damaged mitochondria, like shown here. So normally the process is very fast. Autophagosomes are quickly formed, they're quickly fused with lysosomes, where the content of the autophagosome is digested and recycled. And actually, in a normal cells, autophagosomes can be barely seen. However, if there is a problem between of fusion between autophagosomes and lysosomes, then obviously autophagosomes begin to accumulate and pile up. And that's exactly what happens in Pompe disease. In order to visualize those structures, those autophagosomes, what we did was we made another mouse model of the disease in which these structures, autophagosomes, are now labeled with green fluorescent protein. And we call those mice green Pompe mice, and as a control, we obviously made green wild-type mice. So this is the live muscle fibers from these mice, and as you can see, in the wild-type mice, autophagosomes, which are now green, are seen as tiny little dots. And in contrast, in green Pompe mice, you can see again a massive autophagic accumulation in the core of muscle fiber. So what are the consequences of autophagic accumulation within the cells? First, those non-contractile inclusion may reduce muscle force. And we actually have some experimental data confirming that this is indeed the fact. As I mentioned before, the autophagic pathway is designed to eliminate some of the unwanted products within the cells. So when the process is not efficient, then those unwanted products accumulate in muscle cells, and they potentially may be toxic to the cells. And again, we have the data showing that that's indeed the case. And finally, the presence of this huge autophagic accumulation may interfere with the delivery of the recombinant enzyme. And we now have a direct evidence that this is indeed the case. What we did was we labeled recombinant enzyme, the drug myozyme, with the help of people from Genzyme, and we injected it in our green mice to see where the enzyme ends up. So the enzyme is now red, administered into our green mice. And as you can see, the bulk of the enzyme, the red, ends up in the autophagic area. This is not totally surprising, because it was shown many times before, and here at the meeting, you are familiar with this pathway. The enzyme traffics to the lysosome through the endocytic pathway, and it goes through a series of vesicles within the cell. Early endosome, late endosome fuse with lysosome, thus providing the track to deliver the recombinant enzyme into the lysosome. The problem is that these vesicles of this autophagic pathway and the endocytic pathway, and the autophagic pathway communicate with each other. and autophagosome fuse with late endosome and even early endosome, resulting in the formation of an intermediate structure called amphisome. So it's not surprising that the accumulation of these vesicles result in diversion of the enzyme away from lysosome, and the enzyme becomes trapped in this autophagic area. So what we thought was that if we genetically suppress autophagy in our Pompey mouse model, we may facilitate the effect of enzyme replacement therapy. And what we did was we inactivated one of the critical autophagic gene. The protein which is encoded by this gene is called ATG7 protein, is critical for the formation of autophagosome. If this protein is absent, then autophagosomes would not form. And what we found in our mice, that this genetic manipulation by itself resulted in a significant reduction in glycogen level in skeletal muscle. And what was most exciting was that when we treated these mice with enzyme replacement therapy, we observed near complete glycogen clearance, an outcome which we have never seen with different approaches to therapy. And so this complete glycogen clearance was also confirmed by the staining of muscle section for glycogen, and as you can see in untreated mice there is still glycogen as shown by little purple dots, and this muscle looks completely normal. So for the rest of my talk, I will try to show you muscle biopsy from patients with different forms of the disease. And I would like to tell you how we process this muscle biopsy. We isolate single muscle fibers, and we stain the fibers for autophagosomal marker and lysosomal marker. So now the autophagosomes and lysosome would be colored differently. Autophagosomes would be green and lysosomes would be red, or the other way around, depending on my mood at the particular day. So we then analyze those fibers by microscopy, which allows us to go through the whole depth of the fiber. And I have to say that for each patient, we analyze at least 100 fibers, and in many cases, hundreds of fibers. And I will certainly show you just the representative images. So by using this method, this is how control muscle fibers look. You see in green lysosomes, and they're very nicely and evenly distributed through the whole length of the fiber. And you basically don't see red dots, autophagosomes, because, as I said, they are quickly engulfed by the lysosomes and recycled. So a little red background is a non-specific background staining, so you should look for the structure. I think they need to turn the light for almost the rest of it. Yeah. So what we see in late onset patient, in adult onset cases, we see a striking autophagic accumulation in the core of muscle fibers. In all those cases, you see the accumulation of autophagosomes and lysosomes constituting autophagic buildup. And what is remarkable about those biopsies is that it's even more than in mice. The autophagic pathology constitutes the only pathology in many fibers, because we don't even see expanded lysosomes in the periphery of the fiber. Can I still have a little bit of light?
There's a little bit more. There's nothing in between.
Okay.
It's either this or it's down.
I'm totally in the dark.
If you feel scared, you call me in.
Okay. Okay. Autophagic accumulation is also the predominant pathology in many fibers in childhood onset disease. Here is the two cases of a 7-year-old and a 5-year-old, and there is again a striking autophagic accumulation in muscle fibers. There is yet another way of looking at it, and you can see those little bubbles again showing the presence of autophagic buildup. The autophagic pathology can be observed in these late onset cases very early, as shown here in a 5-month-old patient with late onset disease diagnosed through newborn screening. Autophagic pathology persists on therapy, as shown here by using a different way of looking at muscle biopsy in a 5-year-old patient on therapy since 16 months of age. And autophagic pathology persists on therapy even if the therapy began very early, as shown in this case in 8.5-month-old patients with late onset disease who started therapy at 2.5 months of age. We were quite surprised to see that in infants, untreated infants, the autophagic pathology is practically absent. Here, lysosomes are shown in green, and as you can see, muscle fibers are filled with largely expanded lysosome, which coalesce together, they come together, and may well break at some point. Here is an interesting case, and a 1.5-month-old infantile patient. And what you see here is relatively well-preserved muscle fibers in biopsy with very typical lysosomal pathology, so characteristic for infantile but not late onset forms of the disease. And what I see here that in 20-25% of muscle fibers, there is a classical autophagic buildup, which is so characteristic for the later onset form of the disease. And it turns out that this is a questionable case, that it's quite possible that in this particular case, the patient is suffering from a little bit milder late onset disease. We also had some muscle biopsies from newborn screening program from Taiwan after 6 months on enzyme replacement therapy. And unfortunately, the autophagic buildup emerges on therapy even in infants, as shown in these two cases, with in 6.5 months of age, patient at 18 days at start of therapy, and in 9-month-old patient who started therapy at 3 months of age. Here is a 7-year-old patient on therapy since 6 months of age. And this particular patient responded reasonably well to therapy, but then began to deteriorate. And again, I see huge area of autophagic accumulation. And the question is, could it be that sometimes observed clinical decline on therapy could be associated with the expansion of autophagic buildup? And I would like to finish on an optimistic note and show you 2 cases in which I've observed, at least from morphological perspective, a remarkable reversal of pathology. The first case is an infant, again, with typical pathology for infant, lysosomal expansion, no autophagic buildup, 6 months after a remarkable recovery. But the therapy began in this patient at 5 days of age. And the second case is a patient who started therapy at 1 month of age, although his genetic defect would predict a late onset form of the disease. Again, very mild lysosomal pathology, shown in this case, lysosomes are in red, no autophagic buildup, and remarkably normal skeletal muscle fibers 6 months after therapy. So my conclusion is very simple. Autophagy needs to be taken seriously, and the best way to deal with it is to prevent it. Thank you very much.
thanks so much. It's not only beautiful, but it's extremely difficult to do these types of things. Dr. Raben is actually the only one studying this particular issue in Pompe disease. We will have
others are welcome to study. Others are welcome to study. Other people are welcome to study this.
Lack of time, but yeah, please shoot. Thanks. We will have questions and answers after. So the next speaker is Dr. Priya Kishnani. We do the technology first. You want the mouse? No, this it's on here.
That's fine. I'm just afraid this will...
Yeah, that should be okay. Yeah, should be okay.
So good morning, everyone, and it's a very hard act to follow after Dr. Raben, but I'm going to try and share what I was asked to talk about today, which was the immunological aspects in the treatment of Pompe disease. And I think what I first want to point out to the group here is the package label for Myozyme, and if you read carefully, it says that the infusion reactions are more common in patients with high antibody titers, and then there are patients who do experience infusion reactions. The next important thing that's already stated in the package label is that high and sustained antibody titers, including those who possess 2 null mutations, have a poorer clinical response. And then the next one is that those who have the sustained titers more than 12,800 may have a poorer clinical response to treatment or may lose motor function as antibody titers increase. So I think there was a lot of suggestions right when enzyme therapy with Myozyme was approved. And then when you look at the chart of the treatable lysosomal storage diseases with enzyme therapy, and if you just share or look to the right, you can see that the majority of these therapies, the patients do make antibody titers. This does not mean that the antibody titers persist, but it means that when a therapeutic protein such as Myozyme or Lumazyme is administered, patients will make antibody titers. Now, the part is, when you look at Pompe, about 90% of individuals with Pompe disease across the spectrum of disease do make antibody titers. But does it mean that all of them are going to continue to make antibody titers? I think that's the part that I want to discuss about today and provide some reassurance. Now, if you look at the minimum recommended schedule of assessments for the Pompe registry, even in those days, if you look here carefully, it says antibody testing recommended for all patients receiving ERT according to the following schedule: serum sample prior to the first ERT infusion, and then a serum sample every 3 months. In discussing with many of you in this room, and in following many individuals with Pompe disease who have come to Duke University, we find that indeed this is not the case, even for the infants, where there's a lot more data and a lot more work that has emerged of the negative impact of antibody titers. So I now want to talk about the types of antibodies and the different issues that we face. Now, there are many different antibodies, but for this audience, let's focus on 2. One is what we call IgG, and the other is what we call IgE. IgE, I want you to remember, is like when you have a bee sting allergy. That's what triggers an IgE response. And the IgG is typically what, when you've been exposed to a protein, like say Myozyme, that's the buildup of what we call IgG titers. Now, if someone's having an acute reaction to the Myozyme, they could be mounting an IgE response, which is very different than having IgG titers. So I think that's the first important thing to talk about. So the first issue really with a therapeutic protein like Myozyme is that when an enzyme is infused, people can develop a reaction to it. So these are what we call IgE-mediated or complement-mediated reactions. These are allergic reactions. Some of them can be very simple, like just having a skin reaction, but in some situations it can be life-threatening and lead to what we call anaphylaxis, where there's swelling of the tongue, difficulty with breathing, and actually it can be very dangerous. So this paper we had recently published was on the approach to what we say desensitizing individuals with Pompe disease who've had significant reactions to the myozyme or the lumazyme. And we published this on the infants and children, but we have similar experience now with several adults with Pompe disease. And I think in talking with many of you here, I know you'll have expressed challenges with getting the infusion because of significant reactions. So this is the first part to talk about, that they can be desensitized. And desensitization, as we define it, is the induction of a temporary state of immune tolerance to the compound, which in this case is the enzyme, which is responsible for that reaction. How is it done? It's really done by giving increasing doses of that agent, which is the enzyme, over a short period of time, until the total dose that you want to reach is delivered without incident. And then it can be maintained by continuous administration of the causative medication. When I say continuous, I don't mean continuous 24/7, but on a regular basis. So we've shown this very successfully now in several infants and several adults with Pompe disease who I know have come off enzyme replacement therapy because of a challenge with life-threatening infusion-associated reactions, or what we call hypersensitivity reactions. And what I just want to point out is that as with the care of these of our families and patients, these principles really have to be done on an individualized approach. And so this particular paper that we published, I think, has a lot of meaning. And so any of you who in this room have had challenges with infusion reactions which are life-threatening, please try and go back to this particular paper or have the physicians call us. Joanne Mackey, who's here today, has really spent a career as a nurse practitioner in helping families with infusion reactions. So that's one part of the immune challenges that we face with Pompe disease. And now let's talk about the Igg mediated reactions. And I think you saw the slide yesterday from Dr. Chen, where there are many factors which impact response to enzyme therapy. And clearly, this is only one piece of the puzzle. You just heard from Dr. Raben about the role of autophagy. And now here I'm going to try and focus today about the role of antibodies in Pompe disease. And if you see from our very first work that was done, and Andy Amalfitano is here, who was part of the story when the first 3 babies were treated with infantile Pompe disease, as you can see that the first 2 babies here, after an initial response, which you see in blue against the motor curve, you can see what's in red is antibody titers. And as these titers go up, the clinical response starts to go down. We saw this in baby number 2. And then in baby number 3, who did not make antibodies to the enzyme, you can see an upward trajectory of the clinical response to enzyme therapy. So from here came our first understanding of what I think has now become a very common terminology in the Pompe community, CRIM status, which when we started 15 years ago, I must say, I was often laughed at, and people were not very much convinced by the CRIM story, but I think that it has really emerged as a marker in the field. And so CRIM positive, in a simplistic way, is what we normally see in terms of the protein bands on a Western blot, which is done from skin fibroblasts. So this essentially is showing you the enzyme and the mature form and the unprocessed form of the enzyme. Someone who's CRIM positive makes some enzyme, so you see it as a band on this Western blot. And someone who's CRIM negative is where you do not see any of this enzyme. So as you can think about it, CRIM negatives are those who have never been exposed to this protein before, and so they are naive when enzyme replacement therapy is provided to them. So the patterns of immune response in Pompe disease, and clearly this is based on data from the infantile patients, but I think it's very true, not just for even late onset Pompe disease, but I think it's across the field of therapeutic proteins, that one response, which is the ideal response, is when you make no antibody titers, and so you recognize the antigen, which is the enzyme, as self. There's not a problem. The second, which is a desired one, is where you've been exposed to it now, but now you recognize it, and the body starts responding, but then over time it has a downward trend or has learned to tolerize or take this antigen as self. The third one is this, where you have this high sustained antibody titers, which we see in a small proportion of patients, and I'll try and show you expanding data on this. But this is not just in CRIM negatives, we're also seeing it in CRIM positives, and we're not only seeing it in infants, we're also seeing it in some adults. So I think the take-home message is that there are extremes, but there is a variation to this theme. So from here, we had published our first work on the role of CRIM, and as you can see, if you just look at survival plots, you can appreciate that people who are CRIM positive do better on a survival curve than individuals who are CRIM negative, who by a median age of about 27 months are either completely ventilator dependent or are deceased. And one of the things, and we corrected for this by looking at the age and trying to take all patients who were younger so that we had corrected for confounders. You can see a big difference between what we call CRIM negative and CRIM positive, where in the CRIM positives, they have tolerized to the enzyme therapy, whereas the CRIM negatives have continued increase in the antibody titers, as you can appreciate by week 64. So this is a glimpse at a CRIM positive with a low antibody titer who then tolerized. So this is him at baseline, and then this is him at about 9.5 years of age. You can see that this child has done extremely well, so had the advantage of young age of start of therapy, did not mount an immune response, and has continued to do well. Now, look at a CRIM-negative baby, also started at about 4 months of age. You can see has some preservation of muscle function, but then if you see that at 9.5 months of age is already ventilator dependent and has minimal movements of his extremities. Now these are really landmark individuals because these are from the very, very first clinical trials that were done on enzyme replacement therapy at Duke, and a lot of important lessons that were learned. So now the challenge of antibodies in Pompe disease and what is the extent of the problem, I think we've understood this for CRIM negatives, but I think that there are some CRIM positive patients who also develop the high sustained antibody titers. So how do they do clinically? That's the question. What does this number mean to me clinically? And are they similar or are they different than the CRIM negative patients? The reason why we've picked the infantile population is because the results are fairly dramatic. If they do well, you see them doing well, and if they do poorly, it's very easy to see a clinical decline. It's more of a challenge in late onset Pompe patients where I think we need better tools to monitor the clinical response, especially subtle changes in terms of a response to therapy. So we looked at 40 infantile Pompe patients, and because of the interest of time, what I'll try and tell you is that we separated them into CRIM negative, which we know are a high risk group. Then we looked at the CRIM positive patients, but those who made high titers. And then we looked at the CRIM positive patients who made no titers or low titers. And what you can see is here is the CRIM negative, and the cutoff bar that I put here as the red dotted line is actually even higher than what's in the myozyme package insert. So this is 51,200, so a very high bar. And so these CRIM negative patients have all made high sustained titers. Now you look at the low titer patients who are CRIM positive, and you can see that they're all below that bar. And now you look at the high titer CRIM positive patients, so these are CRIM positive, and you can see that they're very similar in terms of numbers to the CRIM negative patients. So what does this all mean in terms of clinical outcome of these patients? And so if you look at the CRIM negatives versus the CRIM positive, I just want to point out that the green is the CRIM positive, and this is a survival curve of ventilator-free survival. And these are the CRIM negative patients, and these are the high titer CRIM positive patients. So you can see that the high titer patients are really very similar to the CRIM negative patients. So telling us as a proof of concept that having persistent high titers is not a good thing and can actually result in a clinical decline. So what is the percentage distribution of these high sustained antibody titers? Is this really an issue, or are we just dealing with a handful of cases from which we are learning lessons? So once again, this is based on clinical trials data, but now we have a paper coming out on our experience with over 250 cases talking about the experience of CRIM negative versus CRIM positive. And what you can see here is that the CRIM negative and the CRIM positive high titer patients account for about 36.5% of cases cases. This is, of course, for infants with Pompe disease. I think the data for late onset is still emerging. It's not very clear because we don't have very good clinical correlation, but clearly in the infant population we have learned this, that it is a significant issue. It's about 40% of patients of infants can develop high sustained antibody titers. So the next question is, do late onset patients develop IgG antibody titers? And this is really from the work that was published by Ans and by the whole team that worked on lots, is that a number of patients have developed antibody titers. The good news is that a number of them, 61% of them, started to have a decrease in the antibody titers by the end of the study. But if you look at the data, there are 9 patients.